of the class and looking forward to talking about things that happen after we do the whole growing process today. So a whole bunch of topics, and we're going to go through them in this order. So Adil and I will be here presenting. But first, we can do some announcements. Maybe Emily, do you want to run through these?
So I know a lot of people couldn't make it to yesterday's talk. That had to be moved quite suddenly, but I will be sending out the recording and slides for yesterday's presentation later today. I have some editing to do with that. We have 2 homeworks for you, but the one that's related to the project is there's going to be a second progress report due tomorrow at midnight. And we just want from that sort of a progress update of where your project is so far, your stage of completion, as well as these things listed for you depending on which option you've taken. So if you're doing a fermentation or recipe, we'd love to see pictures. Just make sure you're on your way. If you're doing a review of the paper, then please have your outline. And if you're making a paper proposal, then include at least 3 to 5 professors that you've thought about that are relevant to your work, who you could come up to asking to do a research with, as well as a short explanation of why those professors are relevant. And this doesn't have to be super long. All of this progress report can be like half a page to 4 sentences. I just want to check on how you're doing. I'm not giving you more work.
All right, so yeah, we're looking forward to it. Let's go ahead and get started with downstream processing. So again, downstream processing and pretty much all of the stuff we're talking about today is just, we've done all the growth stuff up to now, and how can we actually bring that to our plate? So first, we can talk about cultured meat, and albeit there's going to be a little bit less information on how we do the downstream processing from cultured meat side, just because not as much of it has happened and brought these products to our consumption. But we had a really good lecture from Jasmine on scaffolds and a pretty great one from Emily yesterday on bioreactors. So we can put those together. We can put the scaffold inside of the bioreactor first. And what's actually going to happen is the cells will start to seed on the membrane of the scaffold and actually give us a texture that's similar to this sort of fibrous or like chewable texture of a steak or a whole cut of meat. So that's one way we can actually put the scaffold and the bioreactor together. But another way that we can do it is we can actually, instead of growing our cells in scaffolds themselves, we can remove the cells using a process called trypsinization. So it's really just an enzyme, and it acts like a pair of scissors that can really cut any of these adhesion proteins that the cell is sort of sticking to when it's growing. And from there, we can get this sort of pinkish paste, as this graphic is calling it. And that cell paste is really something we can use as a medium for other types of structuring processes. So when we make something like ground meat or minced meat, what we can do is we can add some fats or emulsifiers and be able to press the meat, the cells, into sort of traditional ground structure. And if we wanted to use a scaffold again, we could also do that on the outside. And so what we'd actually do is we'll take the scaffold and put it in the incubator, and then we'd submerge it in a cell solution of that pinkish paste. And what's actually going to happen is some types of cells may be better at just growing their initial stage in the free-flowing perfusion bioreactor, or in some types of cells might just want to have that really structured adherent ability in the very beginning. And that's just kind of based on which type of cell we're trying to use. So yeah, these are 2 kind of ways that we can process the cells that are coming out of the cultured meat process. But really, kind of a more elaborate thing we can learn about today is what has to do with precision fermentation. And a lot of this downstream processing has been done for a while for human consumption. So today we're going to take a pretty deep dive on what it looks like for Perfect Day, which makes animal-free dairy, but more specifically, animal-free whey. And Perfect Day is a B2B company, which means that they're trying to, I think in the beginning they had started by making their own products, and they currently have like some of their more own product formulations, but they also are able to sell this whey protein directly to other ice cream makers, to other dairy companies, and they're trying to incorporate that whey as a vegan and lactose-free dairy product. So one of the examples of that is this pretty old ice cream company called Graeter's, and they're known for having pretty great ice cream. But now they can actually make their product using an animal-free dairy mix that comes from Perfect Day itself. So yeah, let's take a quick look at that. So when Perfect Day is first coming up with our idea of what do we need to get that creamy texture of milk, they have to think what protein actually is responsible for that. And through their processes, they could find that there's actually one particular protein in whey, beta-lactoglobulin, and that's going to be responsible for a lot of the creaminess, a lot of the emulsification properties of dairy that we're pretty used to. So they've made this the target of their precision fermentation process. And this is a little bit of a review from the crash course. So just how we sort of learned the cut and dry basics of how genetic engineering is working at a really high level. Perfect Day's genetically modified microbe of choice is a fungus called Trichoderma raceae. And when I showed you guys this slide the very first time in the crash course, I kind of just substituted out this whey protein sequence from the one from the cow, and we could just straight put it into the genome of this fungi. But in reality, it's not so much of a simple cut and paste. There are a few things we have to optimize in order for the downstream processing to be a lot more efficient. It's sort of like if downstream processing, we're being able to cut and paste this protein sequence into the yeast or the fungus. It's kind of like we're speaking the language of the fungi. This is sort of like the dialect of the genetic engineering that we're doing. So a few things that we would have to think about here is basically anything other than the whey protein sequence itself. So that protein is going to be pretty intact in terms of its sequence, but things upstream like the promoter, terminator, the whole cassette, as we call it, there are some ways that we can make this more effective and more efficient in the fungus itself. And so we're going to substitute those parts out, and the actual sequence will be native. Okay, so Perfect Day's methodology, this is actually taken from their GRAS notice, what they actually had to submit in order for their product to be approved and sold as a food additive. This is a very high level, and we're going to go in through it. You guys are going to be seeing how we go from the fermentation process to the actual purified protein. And so the first is just sort of a seed train of we've genetically modified our yeast, and we're going to put it into successfully larger bioreactors until we can get a really large amount of solution that we can work with. And then when we have our huge sort of vat of liquid that has been growing and growing, we can actually check the optical density of the solution. And what that's going to tell us is just how much yeast or how much microbe has proliferated in that solution. And hopefully there's enough to the sense that we can say, you know, if we're at this particular optical density, we can expect to get this much product out of it. And from there, what we're going to do is a solid liquid centrifugation, which just means that we're going to spin this down in a centrifuge, and all of the really big things in the solution, based on the time and the speed that we're centrifuging at, they're all going to collect in this thing we call a pellet. And the special case of Perfect Day and the protein that they're trying to make, whey is a soluble protein. So the fungus, when they're in the solution and when they're reproducing, what they're actually going to do is they're going to secrete the whey protein into the solvent, into the media. And so we don't actually need to break open the fungi at all in order to get our product, but it's actually going to be secreted into this rest of the solution called the supernatant. So we can empty off the supernatant just by pouring the jar and get some clear liquid that looks like this. And to actually get the whey from that clear liquid, what we can do is we can change the pH of the solution, essentially to make the protein not have a charge. And when the protein doesn't have a charge, it's going to clump together because the attractive forces in the protein are really just going to encourage it to stick to each other. And we can actually see that process usually happen. And we'll get another clump, but this time we want to keep that clump. And there are another number of ways that we can actually separate that clump out from the liquid. One really popular one is called chromatography, and essentially at a very high level, it's just we can filter off this liquid through some beads or some sort of filter that can manage the size, the charge, and other properties and pour them out in a certain order. So for example, if there's a mixture of large sizes of particles in a solution, we can expect that the larger particles are going to go through the filter a lot slower than the smaller particles. And we have some pretty sophisticated computers and machines that can tell us exactly which fractions of this filtration solution are the ones we want to keep. And then from there, when we have just that pure solution, all we have to do is freeze dry it into a powder. And this is actually a picture of the product that Perfect Day makes. And that powder is what they sell to their B2B vendors. And so yeah, it's pretty crazy to think that we can now understand how we can actually change the DNA and also get the product that we want. But now that we have the product, how do we actually ensure that we can use it? And as a very, very, very basic step, there was a question about this, I think, on the first day, which is why I bring it up. But really in any sort of consumer goods manufacturing, there's a set of principles called CGMP, which basically just means that we have to adhere to these. And there are things like, keeping the workspace clean, making sure it's sterile in terms of we're not going to have vents bringing in random poisonous gases into where we're creating these things. So this is a pretty standard and basic protocol we'll follow. And this is kind of when I talked about precision fermentation and how we can regulate these, how we can actually bring the products as food additives through the FDA in the US. One word that I brought up was GRAS, or I guess it's an abbreviation, but generally recognized as safe. And so just as a refresher, what this means is we can actually have a third-party company come in and evaluate our product. We don't actually need to wait for that singular team on the FDA to come in, get our product, test it, and give us a result. But by having a third party come and do it instead, we can just send those results in a report to the FDA called a GRAS notice. And then the FDA can either say, we have some questions or we don't have questions. And that no questions is kind of the sort of indirect approval that you're getting to be able to put this in the market. So an enzyme that I talked about on the first day as well, called chymosin. This is actually what was used instead of Rennet for cheese curdling, and Rennet is originally sourced from the inside of a cow's stomach. So instead we found, can we make this using microbes? And chymosin was this enzyme that we could actually make using microbes instead. And what's so special about it, it's the first enzyme that was approved for food by the FDA that is microbially produced. So this is really one of the hallmarks of precision fermentation as a practice. And this kind of had set some of the precedents for regulation in terms of, an enzyme that's actually coming from microbes is different than an enzyme that comes straight from the animal source. And there are some criteria we have to think about for why it's different. Things like from the manufacturing process or from the media might be in there. And we need to check for these things to make sure it's still safe. There was okay. Yeah. Rennet, I believe, is the correct pronunciation. Thank you, Adele. Yeah, so yeah, we can think about just how some of this regulation is being modeled and being shaped from this initial one that was passed in 1990. There actually came up with a kind of decision-making tree as to how we can think about the regulation of food enzymes in particular. So these two people, Parisa and Johnson, in 2001 came up with this kind of decision-making tree. Yes, it's genetically modified, but how stable is that modification? How dangerous does the potential have to be? How consistent are the products? That kind of thing. What is the total observable effect, adverse effect level that we can tolerate, which is actually what this NOAL stands for. But back to Perfect Day. their GRAS notice was actually developed from 2018 to 2019. And, you know, along with just the basic stuff of saying this fungus has been used in production before, we know the kind of toxins it's able to create, and we're able to screen for those. We're able to show that wherever we put in our DNA, it's going to be consistently producing this product, and we don't have to worry about it being jumping around or being randomly inserted. And really an important part of their analysis is also to show that, you know, in the first place, their microbial way, which is this ILGB, the beta-lactoglobulin in this column, is a similar size and similar charge to the whey that usually comes from cows. As you can see, these bands are in the same place in this gel, which basically just means that they are the same charge or very similar size and charge. And lastly, the kind of third party that was involved in this GRAS notice was the University of Nebraska at Lincoln, and they were actually able to run an allergenicity analysis. So not just showing, you know, we're getting the same amount of whey in each sample, but we're also able to see just what parts of the microbial process are left behind in the solution. And they actually found that the highest level in all of the solutions they tested, I think they have to do something like 5 batches, was like 6.7% of just structural protein from the fungus. And they concluded that this wasn't enough of a level to actually cause any sort of allergy in people, which is being able to make that kind of analysis is pretty important to approve this. So the FDA read all of this and they said, no questions. And that is why we're able to enjoy Perfect Day today. So I just wanted to jump in pretty quickly about some kind of hallmark regulation or places around the world that we have to think about in terms of how they're regulating these things other than the US. And one kind of interesting place for sure is the EU. In general, the attitude towards GMOs or products such as those produced by precision fermentation are a little bit more reserved in the EU. For example, BT11 is a variant of corn that can be resistant to a certain type of corn parasite. And so in the US, we can actually approve the corn. We can approve any product really that's used for environmental or human consumption at separate levels, and they're kind of independent of each other. Whereas in the EU, we're going to have to first approve something for an environmental agency, and only after that can we think about using it in food industry for human consumption. So kind of differences in the way that it's structured in the approval process. And possibly some precedents for why there might be a little bit more reserved attitude towards GM products or just even new foods in general. In 1986, which is also sort of around the time we're starting to see a shift to developments of these technologies, there's a pretty bad outbreak of mad cow disease in the UK. And from there, one of the reasons cited for why these cows were getting so sick and possibly even transferring this to humans is because one of the cited sources from observers was that the industry was producing beef by feeding up ground dead cattle to live ones. So they're effectively turning herbivores into carnivores and carnivores into cannibals. So this was kind of frightening to people in the sense that the industry sort of was tied to this idea of having motives other than just feeding people, but trying to find cheaper ways to feed people and feed people in that way. So with the rise of GM technology, it was kind of thought that possibly, even though there's no link between mad cow disease and GM products, that there are other motives at play, and so a little bit harder to adopt these kind of technologies. And yeah, another sort of interesting feature of the regulation system is that there's case-by-case regulation. So actually, the European Food Safety Association, effectively very similar to the FDA here, they actually have to go through each food and be able to look at it themselves. So it's a direct approval process rather than us where we can just say, hey, we want to go through the GRAS process, we're going to have a third party come in and look at it. But eventually, things like the need for food security and having imports and things like that, there has been kind of a shift in attitude, it seems, through citizen survey. Yeah, and then a complete other end of the spectrum, Singapore, as we've talked about before, is a country that has rely a lot heavily on imports for food. And so they actually have something called the 30 by 30 initiative, which is the goal to produce 30% of their own food in Singapore by 2030. And so that's where we're seeing a lot of regulation being coming up on novel foods. And particularly, this is actually taken verbatim from their section on cultured meat. So this is how Just Chicken was actually approved, came through. And this policy has been in development, I think, since November 2019. But Just Chicken was only approved on December 2nd of last year. And so, yeah, some of these factors are things we've talked about in this class. And it's pretty interesting to just think that people who are running the government are also thinking about these things. Yeah, and as a very last fun slide from my section, I just wanted to know what was going on in the actual Just Chicken Nugget. And it looks to be about 25 to 50% just that cell paste, so that pinkish solution we showed earlier. And then 10 to 20% is their sort of binder, which instead of an egg, they're using mung bean, which is also the foundation of their plant-based egg. So yeah, a huge reason for the reduction in price in the Just Chicken Nugget looks to be that they have been able to reduce the amount of FPS they're using. And I think originally it was like $50 a chicken nugget, but now it's been able to be reduced to $23 an entree. So things are changing and hopefully getting better and better for this. All right, I'll shift it over to Adil. Do you want to share a screen or do you want me to?
Yeah, I'd want to share a screen. Okay. Right. Can you see it? Yes?
Yeah.
I'm going to go into present mode and, all right, great. So I know some of you guys are definitely interested in entrepreneurship, but not everybody is. So I'm going to really try to make this section a bit broader to include looking at, why or why not you should do entrepreneurship, kind of state of the industry as a whole, and the landscape of some of the current work that's been done, as well as major white spaces, and then kind of just thinking about working at a startup, because currently the vast majority of these companies are startups. So working in this space is kind of, you would benefit from having a bit of understanding and savviness around entrepreneurship in general, and a little how-to on starting your own startup. Okay, so to first start, I want to talk about kind of why not to do entrepreneurship, because this is a really great talk done by Startup School that so I'm taking some of the slides from there, where they kind of try to demystify some of the myths that are out there about entrepreneurship. The first is financial outcomes. A lot of people who are interested in starting their own companies think that they're going to make substantial fortunes from doing so. In actuality, most companies don't make it very far to the end where you'll have a big payday. And during that process, you usually have to raise a lot of money. So the stake that the founders actually have are usually quite small, somewhere in the ballpark of 10% or so. And you'll likely have actually much safer probability of having a good financial outcome that could be decently similar size to starting your own company if you actually join a later stage growing company. So a company that's already worth maybe a couple hundred millions of dollars. And if you work with them till they scale to maybe 10, 20 billion dollars, which is quite a bit of growth. But if you can do that, you'll likely make right around the same amount of money. The other is impact. There's definitely a lot of impact you can get from starting your own company, but there's so much leverage that you really get from working at an established company that has a working team, strong infrastructure in place, and already has a user base and connections that allows you to build on top of the work that has already been done. And can potentially, have much larger impact. Another main myth is kind of the lifestyle is very glamorous and it's fun going out and spending all your time pitching, and in actuality, it's just a lot of hard work and a lot of stress. And you kind of just have to be doing it because it's, interesting and exciting. And and the headlines that you see from people raising a bunch of money, it's just, years of hard work to get there. And there's few overnight successes. And then the degree of agency and control that you feel that you think you might have, where you think you might be like calling all the shots if you found your own company. Usually it's the case that you're just trying to put out a lot of fires that are happening. And so everyone else is kind of your boss in that way. So these are some of the myths, and I know it's a little bit of a downer. So that's not to say that you shouldn't do entrepreneurship. I'm just knowing what you're getting into, which is really next point, and the reasons why you should be doing entrepreneurship. So is really, you can't not do it. It's a mix of really having a strong passion for the thing that you are working on, that you're going to want to do, because it's going to be a long time and a lot of years of work, and with no guarantee that you are going to actually be successful at the end of the day. And the other side of it is that you have a strong aptitude specifically for the area that you're working on. So if you're one of a few people who have really good expertise in a specific area or insights and experience, that makes you well qualified to kind of execute on the thing that you want to do. And a good analogy for this that I've also heard is thinking of it kind of as becoming a professional athlete, right? There's no guarantees at the end of the day that you're going to make it. There's a lot of things that are outside of your control, but if you're really passionate about what you're doing and you have some skills if you have some skills that give you a strong edge, you can potentially go the distance. So now I'm going to kind of shift into more about what the field is and what it looks like right now. And this is from the new protein maps that are put out by Olivia Fox Cambon. And it's so you can see here, there's a lot of startups that are already out there from on a wide range of things. And even in cell-based, you have air proteins, dairy, beef, eggs, multi-species fats, gelatin, growth media, and several others, and numerous numbers of ingredient companies that are currently trying to, tackle this space and, stake their claim in the new future of food. And it's actually decently spread out geographically. This is specifically a map showing cultured meat companies. A lot of them are based in the US. And I think in no small part is that the US kind of fosters this type of innovation and is willing to throw a lot of money at these type of companies, and especially with good branding. So for example, Memphis Meats, or even kind of, if you think about just in alternative proteins and like the plant-based world of Impossible Foods and Beyond Meat, which kind of really fit the build of traditional Silicon Valley tech in the way that it's funded and the kind of timelines and horizons for execution. Yeah, so for those reasons, building something in the US, you definitely have a lot of advantage, but it's everywhere. And the field also splits down right now into two main groups: B2C startups, which are actually just trying to develop products that can then be sold to a customer, so business to consumer. That's what B2C stands for. And these are kind of breakdowns, the wheels, and I already went over them in the other map, but it's nice to kind of see them all laid out. I also want to highlight, at least briefly, pet foods, because I feel like people don't really look into much about pet foods. It was one of the kind of more interesting markets that I think is less explored. Recent figure I heard is like, it's somewhere between, it's in tens of billions of dollars as a market, and somewhere between a quarter and 30% of all greenhouse emissions that come from foods are actually associated with pet foods. So there's a lot of impact that can also be made in that space. And then there's the B2B companies, and these companies are much more recent. And it's a byproduct of just the growth of the B2C industry for cultured meat, that these companies have been able to survive. And there's a lot of stratification. And that's, I think that's the real key here. You can see that there's people who are developing some bioreactors and cultivator designs, scaffolds, cell culture media, and then cell line development. And that, and the expectation is kind of that the industry is going to have a decent bit of stratification, but and at least some of the earlier players in the B2C, like Memphis Meats, for instance, are really doing, or Mosa Meats are doing a lot of vertical integration for having kind of the whole pipeline built internally. But for a lot of these other small fledgling startups that are out there, this stratification is really going to be the backbone of how they grow. So the next main one is that I wanted to talk about is a bit about what's the breakdown in jobs that people might want to look to. These stats are a bit older. This came out in September 2018 from some nice blog post that Wildtype put out. Wildtype is a cell-based seafood company. And they did a bit of analysis, and right now 80% of the current non-founder roles are scientific, or September 2018. And there's expectation that actually this is going to shift quite a bit as the area grows. So for people interested in non-scientific roles, there's going to be a lot more growth in those specific areas. So you're talking about the HR, business development, operations, marketing, and communication. There'll be a lot more growth in them. But of course, plenty of growth in the science roles as well, but likely not the 80% range. Yes. And then kind of the size of the investments that have been happening in cultured meat. So in 2016, it was a much smaller space, and it's really been exponentially growing overall. And the number of deals are numbering in, the low 20s, with tens of millions of dollars being put in annually. And kind of a bit more of a summary. This is up until right around the end of the first quarter of 2020. There's been cumulatively 166 million that's been put in, with I think close to half of that just coming in 2019, and the largest investment in 2019. What's interesting? Blue Nalu, I think you pronounce it Nalu, I'm not sure, just raised, actually, they announced a couple of days ago, another $60 million round. So there's been massive amounts of funding that's been keeping these guys together. And a lot of them, so the majority of these companies have just been doing Series A's of around 10 million. And now a lot of Series B's are coming in. Yes, and COVID was in no small part accelerating the pace that these companies are working at, as there's a new spotlight getting put on the future of food with the current infrastructure and logistics around how our food system works, really showing its constraints during COVID. And this is a more cumulative chart showing back all the way to 2010 for a breakdown of all alternative proteins for fermentation, plant-based foods, and cultured meat. And now this also interesting cultured meat that we talked about looks so much smaller in the grand scheme of all of the rest of what alternative proteins happening, but it's clearly having exponential growth. But I would say a smaller rate than what fermentation has been doing. Fermentation has been really killing it. And at least in terms of the kind of areas to be looking into and working on, fermentation is definitely the most promising, in my opinion, for reaching to market, just because of how much more established the current infrastructure is that's out there, and the current products that are already serving customers. And then, of course, plant-based foods have been around for a while and are growing more and more. And definitely shouldn't discount them, because a decent bit of the ways that people will be eating food in the future will just be plant-based. And that summary, you know what, I'm not going to get in too much into all of these numbers, but you can look at them. Basically, the main ones to highlight are really the largest rounds being like 140 million. I think this was Perfect Day. Just in 2019, there was 247 done, and nearly $1 billion between 2013 and 2020. And just also a comparison between 2019 and just the first half of 2020, it's like 437. This is just for fermentation. So it's pretty crazy, very global as well. Okay, and so now kind of into major challenges and interesting white spaces for clean meat. Specifically, I think this is a really nice mind map that shows a lot of the different topics that we have been going to over the past couple of weeks, kind of all put into one figure. So you have culture media, where a lot of the main developments are really on trying to reduce costs and optimize them. Then there's bioreactor design. How do we harvest, filter, and allow for sensors and other types of tunable controls on next stage bioreactors that allow for high quality growth of these cells. Then there's cell line development, which I think Emily did also like a deep dive into. So cell lines are highly important because they're bringing the taste, flavor, and also if you can get it right, immortalization strategies make less reliance on having to continuously get primary cell lines from animals. So there's a lot of work that's being done across there. Scaffolding and structure, which I think Jasmine talked about. There's several different methodologies. This one's kind of a bit farther down the line, but an interesting still kind of a white space. I'd say at least in order of the amount of work that's being done, is most of the work I think is really focused on cell culture media and cell line development, with definitely a bit happening in scaffolds and bioreactors, and I would say not so much happening in supply chain and distribution, mostly because those are kind of concerns that are a little bit farther down the line. And so as you can, the organization of a lot of how things have been done are just what is the most immediate. And if you are really betting on the space growing and long-term being as big as you think it's going to be, then all of these other constraints, such as how do you do supply chain very effectively, or how do you do scaffolds to have more structured meat products and not just unstructured kind of mashed food, is going to be really valuable and something that you can target. And similarly, you can also look at fermentation in the same light, but it's some of the concerns are a little bit different now. So now a lot more interest, instead of media formulations, it's the wider space is like kind of feedstocks. So what are all the components that are needed to sustain the cells? Approaches, which for the vast majority of what we've been talking about has really just been precision fermentation, but there's plenty to be done in traditional and biomass. For organisms, there are a number of current organisms that are currently being used for productions of different food additives and are approved for those. So you can kind of look into the biodiversity of those, but developing really good scaffolds, or not just scaffolds, but what are called like chassis that you can then produce the compounds of interest effectively, cheaply, and reliably, and with other value adds like secretion, for instance, like Sangeeta was talking about, are really important. And each organism kind of has its own pros and cons that, depending on the products that you're trying to make, will really define which ones you want to go after. So there's, I think, also with the development of new gene editing technologies and sequencing capabilities, we have a nice workspace of making custom chassis for each of the bespoke products that we want to make. Then there's a series of different services. And okay, I'm not going to get too much into this, but different services of things that can be done with fermentation, production methods, product types, and business strategies. Yes. And I think this is the last one on the major challenges and interesting white spaces, is at least when talking about the overlap between different components in alternative proteins. Plants and plant-based and cellular-based are kind of meeting in the middle right now, and there's a transition that's happening where a lot of work that's not just tofu, this tofu is an example, not just a lot of work that's being done in making high-quality plant-based meats can and are benefiting from fermentation as a strategy to produce additives that enhance the tastes and flavors of these plant-based, which shifts it more kind of into a hybrid model, which is where Impossible Foods kind of lies. And then it's kind of a sliding scale to then going fully cellular, fermentation derived proteins, different components, and also just cellular-based food, so clean meats. Okay, and on this sliding scale, there's a lot of opportunity where most of the work that's a lot of work's being done here, and more and more work's being done here, and there's along this gradient, there's space for improvement that not as much has been happening in. So some highlights are like Motif is one company that's trying to develop really good additives, but yeah. Okay, and I forgot this slide too. So this came out in another great post by Wildtype. This is just another visualization if you try to think about the whole process of how things are getting done. So going from cell line development, formulation, cultivating, actually texturizing it, prototyping it. So these are kind of stages of what it takes to like bring a product to market, specifically a clean meat product to market, and all of the white spaces are areas where there can be work done. I think this one's also came out in 2018, September 2018. And I think since then, there's been definitely more work that's been looked into for facility development, but it's low. So I don't know. This actually holds true for the most part still. And a lot of this, as you can tell, is really about supply chain development than it is about really a ton of scientific concerns, but very valuable to consider. Okay. After all of that, so kind of understanding a bit more about the space and entrepreneurship and what has been developed so far, I think a good framework or at least points that you want to consider when you're looking at a startup to work at specifically. That a lot of these teams are going to be quite small. So really looking at who's in this team and what is their background, what have they accomplished in the past, and why are they like good people to then work under, as well as degree of culture fit, the problem that they're really trying to solve. So what's happening is that if you go back, if you remember back to the map of the space that I showed you, there's a lot of overlap that's happening, and there's people who are kind of doing the same things that other people are doing as everyone's kind of just seeing this valuable opportunity. It's a bit of a gold rush. And so what exactly is the problem they're trying to solve, and how are they solving it, and what differentiates them really strategically that from the other main players? It could be that they have a new technology, or it's that they are geographically a new place that has different concerns. It can be leveraging regulatory things, or since the space has grown so much, they're trying to provide different services and equipments that others are not looking into. And the other is funding. So how much funding does this team have? Kind of gives you a sense of one, how much more growth there likely will be for this company, and so how much you will end up getting monetarily, at least, and also about what's the likelihood of success. So more funding rounds that they have, the less concern that there might be that or things are just going to fall apart. So a lot of these companies are really small right now, and it's not trivial that there's going to be a decent bit of consolidation that will happen, and competition will end up resulting in attrition of company. And so you kind of have to just be mindful of that, and in terms of job security and how long you want to work at one of these companies. So the other are customers. Are people currently using their service, and how reliable are those customers? Competition, I already mentioned, and growth in the space. So there's differences between clean meats versus fermentation versus looking at some plant-based products. And then even within there's all the different stratification I talked about. All of that is growing at different rates. There's valuable to being in a nice white space where there's not really much competition, but how long is it going to be till you have a good customer that needs this service, right? These are kind of things that you have to balance and see for a startup that you're considering to work at. And actually, a lot of the same concerns kind of come into play when thinking about starting your own company. It's really about team, idea, strategy / business model, and funding. So just high level, these are kind of really high level points. And I really, if you're like seriously considering doing something like this, starting your own company, there's a lot of resources that MIT has, the Martin Trust Entrepreneurship Center that's out there where you can learn and gain a lot of valuable insights. But just kind of high level, if this is something you're considering even doing down the line, I really think of starting your own company as a team sport. Don't do it solo. Have a co-founder. It's really rough, and there's going to be a lot of downs. And having somebody who you're working with, it makes it more bearable. The best type of co-founders are friends and people you've known for a long time, people who share your values and you've worked with before. And lastly, if you're thinking about this as something down the line, really network with people. Grow your network, because it's going to be so valuable for people who you might and think about working with down the line as doing something like this. You have to be in a place where you know people who have done stuff like this or you have worked with before. So you have, optionality and stuff that you can do. So even if you're not thinking about doing this right now, if you think maybe sometime in the future you might want to, having a really great network is really important to that. And to that end, college is probably the best place to grow a really high quality network. So look to your left, look to your right. These are some people who could potentially be co-founders with you. So okay, so ideas. So really, again, this is a bit high level for ideas. The things that I really consider as being some of the best problems to solve are ones that you've experienced yourself, know a lot about, or know people who face on a frequent basis, because it gives you access really closely to customer, their concerns, and how to solve them. So to that end, at least, I can give you an example for something that isn't a startup, but is a project that we're at least trying to do a bit internally. If you think about the space of alternative proteins, it's a vastly growing one, and really big concern for companies is actually accessing customers and matching with them in a meaningful way. And for customers, it's really hard to find all of the new great products to test out. And so one project that we were looking into doing as a team internally, just as one of a community project, was how do we make a good listing that's searchable for people to discover really great products and for companies to get discovered. So it's like a little registry that we're trying to do. Example is a problem we face. So we know a decent bit about it, and So also the other things is start with problems. Don't try to start with solutions. You start with solutions, it's very difficult to then connect it back to your best customers. And don't try to force it, because if you really try to force ideas, usually it doesn't end up working out. So things kind of work best when they're a bit organically. So just try to be in the right spaces, thinking about hard, interesting problems, things. You'll generally land on something valuable over time. Okay. The other kind of really big component is strategy and business model. So that breaks down, at least what we've talked about a bit so far, is B2B. And the main playbooks that people are doing for B2B is trying to develop ingredients that are really valuable. So think of kind of like different protein or food additives that people are like Motif, for example, that is trying to give to a plant-based company, or even like Mission Barns, which are making fat cells that would be added to a plant-based food or a clean meat food. So that's a valuable ingredient that can be used. And the other is feedstocks and materials. So media formulations and components that are used to make media formulations. The other is the B2C, which are highly focused on end products and foods that people will end up eating. So think Mosa Meats, who's actually trying to make somewhat of a burger. A lot of actually the B2C companies are also looking into doing B2B with larger meat companies, but for the most part, like Mosa Meats and Memphis Meats are trying to actually sell to customers, and they are making an end product, which is a meat. And then there's also B2C ingredient companies, which are trying to really sell components that other people will use in baking and cooking. So to some degree, you can also kind of just consider this as end products, but it's nice to separate them out a bit. Okay, and then the other kind of business strategies that are not really explored as much right now are B2B services and equipment that is also really valuable. It's just that the space of current companies is kind of small. So this as a business strategy is a bit weaker until you have a larger space of companies, but is also really valuable. And kind of there's innovation that startups can do, not just in the products or technologies, but also in the strategies and business models that they are working under. And then lastly is different types of hybrid approaches to reach to market, where you work both B2B, B2C, one example of that could be Impossible Foods, which worked on actually selling to restaurants at first, and then reaching customers that way, getting name brand recognition, simplifying supply chains with doing a bit more wholesale before they then transitioned over into actually trying to have it in grocery stores. And then the kind of last main thing is like where to get funding. And there are a number of different sources, so I'm not going to try to list them out, but at high level, there's a lot of different incubators that you can apply to. Definitely a lot of research in academia, which will support new work, and nonprofits, which will tend to give grants to people who are working on really interesting companies. Under manufacturing, there's kind of people who, there are divisions within manufacturing companies that will tend to give a bit of funding for companies that will kind of fit into their own business models. Then there's kind of different degrees of VC fundings and corporate partners that you can also get funding from, and a bit of a breakdown of where people have been getting investors in the past. The vast majority, 56%, is from VCs. Then there's another minority of corporate VCs, angel investors, and different accelerators. The smallest is kind of governments and universities, but there's definitely a lot of money out there. And I think that is the end of the presentation, right? Yeah, I think Sangeeta, are we planning on saving marketing for Monday, right?
Yeah, I think we'll keep it for Monday. Will be on food science, and then also a little bit of marketing.
Okay, great. So that was it. Do you guys have any questions?
I think we also have, I wanted to talk about the homework at the end.
Okay. Yeah.
I think there are some slides later. Okay. Yeah, so, all right, thank you. So I did say that we have our second progress report we're asking for you to do by tomorrow, just 4 sentences or about half a page. That's all you need. And then we have had 3 reading assignments so far for this class. So the first one was sort of an overview of cultured meat. So I wanted to give a paper that's also an overview of precision fermentation. And we've referenced this particular paper a number of times, but we'd like you to read the GFI fermentation report before Monday and not read all of it. It's not technical writing, so it shouldn't be too hard to read, but there is a lot of there. So we're asking you to read sections 1, 3, 4, 6, and 7 before Monday in class, and then we'll have our 15-minute discussion on those sections. It's going to be hopefully a really interesting look at with the science and some of the marketing that Sangeeta and Adil talked about today. Yeah, so that's all the homework that we have for you all. Do you have any questions?
Yeah, I was wondering about what kind of services, what the categories of services for the B2B companies means, more or less. I know I saw Emily linked one company in the chat, so that was an example, I guess. But I mean, if you know some kind of examples of this category.
Yeah, the one Emily linked was a great one. Culture Biosciences is basically trying to do bioreactors in the cloud. So you can send them a sample, and then they will validate a bunch of different conditions for bioreactors that would be needed to optimize some parameter of production of some type of compound. Other types of services are people who really work with rapid prototyping and development, so kind of in the same vein as bioculture. So companies that are working on helping other startups on initial media formulations. So not necessarily selling them bulk amounts of media, but actually just helping them get started about what works. Then there's other services that are kind of more in the traditional CPG kind of range, where you're saying, how do you help a new company reach customers? So that's another type of marketing service that that would need to be done. There's other services that look into distribution and packaging. So there's not really one offhand that you can mention. There's a variety that where basically in each one of the stages that a company is going through with their own development, there's potential for a service that can be used to assist in that development process, especially when that problem is faced by several different companies and having a collective solution to it. So one player solving the problem for a lot of people is advantageous, either for infrastructure reasons being involved to develop the solution, or the aggregation of data allowing for higher quality solutions. I think briefly one thing that might help to kind of look at this is maybe if you look kind of down this pipeline of the different all of the different components that companies might go through. You can imagine helping people or innovating as a service along the way here.
Okay. So also things like the marketing side. Yeah. Okay, thank you.
Yep.
Yeah, I listed some names in the chat of people I believe are working on sort of the marketing side, or as well as general consulting, so things like HR, that I would guess is services, I categorize as services.
Yeah. And then the other half of that coin is kind of more like the CROs kind of thing, where it's on the research side. What is like research consulting and services?
What's a CRO?
CROs.
Explain these business terms, acronyms.
The CRO stands for Contract Research Organization, where basically if you want to run some type of experiment, you can basically define the parameters of your experiments and the materials involved, and you can send it to basically the CRO to then run those experiments for you. So you don't have to deal with a lot of the different aspects that are involved of actually running your own experiment. It's really valuable in a lot of, especially for pharma companies, and different CROs kind of specialize in different in different protocols and skills. For example, there might be a CRO that works well at like doing experiments with mice, for example. So if you wanted to test something with mice, instead of having your own mice lab, you send it to them and they'll do it for you.
Or,
yeah,
anything with, I guess, large, or, sorry.
No.
I remember working in Geltor that, we hadn't created our own pilot plant, but we're instead just using a handful of fermentation tanks within a larger facility that I guess rented them out, because buying your own bioreactor, stainless steel bioreactor, as we talked about last time, is a quite large initial investment.
Yeah. So this is another great example of potential service that would be valuable, where there's a lot of companies that are interested in actually producing some compound through fermentation. There's existing companies that have infrastructure of their own that they're using for their own solutions. If you can potentially connect those two parties to find a way where actually leasing out existing infrastructure and for bioreactors is profitable for both parties. It would substantially reduce barriers for new companies to produce products of interest. Right now, the way it's kind of handled is just these smaller startups. Perfect Day is, even a big company now, but it's still, in the grand scheme of things, relatively small startup that they have to kind of work with ADM, and they've just been making really big contracts with different companies that have bioreactor capabilities to produce proteins, the whey proteins and casein to make their milk.
Yeah, there's also the example of when it comes to bioreactors, I think in the very first class we talked about companies that are going in and repurposing and retrofitting ethanol plants to achieve some of these fermentation goals. Yeah, if there's no more questions, there's actually one more slide after the homework, if you wouldn't mind going to that.
More slide after the homework?
Yeah, you might have to exit.
Wait. Exit. Okay, here.
Yeah. So in line with some of the discussion we've been having over scaffolds and bioreactors, pretty interesting talk is happening, I think, next Wednesday on how you can create whole cut meats without scaffolds. And this is actually going to be during the time of our class. So they do post recordings generally. GFI has these Science of Alt Protein seminars, and there's a really good playlist of them. I really suggest checking them out. I've listed them on YouTube below if you have any free time.
So also in this range, we have our business talk, our business representative talk for this IAP happening this week, this Friday. We are going to host Dr. Christophe Chantre of Boston Meats. He is the CEO and the co-founder of that company, which both Sangeeta and Jasmine are working on for texturized scaffolds.